Welcome to Biology Banter.
Today's episode is over cell division and the errors that can occur.
More specifically, cancerous cells and their deformities.
I'm Sophia.
And I'm Lauryn, and we are your hosts.
Let's get started.
To understand how the errors occur, we must explain the cell cycle. We have studied this for the past few weeks to completely understand the topic.
The cell cycle consists of three major phases: interphase, mitosis, and cytokinesis. In interphase, there are three subphases. Interphase is where the cell grows and replicates. The first stage is growth 1. This is where the cell will spend the most time.
G1 is the phase where protein synthesis occurs. Protein synthesis is when the cell makes proteins through transcription and translation. This phase is also where the cell does daily activities. The DNA, deoxyribonucleic acid, located in the nucleus, is in a ball of stringy mess, much like my bedroom, also known as chromatin.
Adding on to what Lauryn said, special protein checkpoints such as proto-oncogenes and tumor suppressors ensure that the cell has completed this stage correctly and is ready to move on to the next phase. This phase is called synthesis. The term synthesis means to build. This is when the cell commits to cell division. DNA replication also occurs, which is when the cell copies all genetic material.
Also, the DNA stays the same as G1, but we see the cell itself grow. Moving on to the next phase, we have growth 2. This stage is known as the resting stage. The cell doesn't do much in this stage and is preparing itself for mitosis. The biggest thing the cell does in this stage is replicate its organelles. Cell again checks for errors in the replication to ensure that the process happens correctly. The cell is ready for the next stage.
Hey, I've got a good joke for this one. What did one cell say to his sister cell who stepped on his toe?
I don't know.
Mitosis! Mitosis is nuclear division consisting of 4 separate stages: prophase, metaphase, anaphase, and telophase. The first phase, prophase, pro meaning early, is when the previously discussed chromatin condenses to chromosomes. Chromosomes are X-shaped structures located normally in the nucleus and consist of proteins and genes. Each cell normally contains 23. The centriole, a non-membranous organelle, appears on opposite sides of the cell.
The nucleus also falls apart or disappears during this stage. While the nucleus disappears, we begin to see slight spindle fiber formation. Again, the checkpoints are reached and the cell moves on. As the cell moves on, we reach the second phase, metaphase. Meta means middle, which will play an important part in this phase. The chromosomes line up in the middle of the cell, and the spindle fibers will attach to the chromosomes in between the centrioles and the centromere, which is the middle of the chromosomes. Once the following steps are complete, the cell will move into anaphase.
Ana means apart, which again happens to play a big role in the main objective of this phase. The chromosomes are violently ripped apart and flipped into V's instead of X's. The previously chromosomes, now chromatids, will also move to opposite poles. This is also when plant and animal cells start to differentiate. In this stage, an animal cell will become elongated and a plant cell will stay the same. Again, checkpoints will ensure that the cell has correctly completed this stage. Next, Lauryn will explain telophase.
In this stage, telo is defined as 2 nuclei reforming. The V-shaped chromatid will relax back into chromatin at the opposite end of the cell. Just like the definition of telo, we see 2 nuclei forming in the cell. This stage is the final step before cytokinesis. It is also referred to as the undoing of prophase and prometaphase's work. Now let's move on to
cytokinesis! Sorry for interrupting.
I think we are growing apart, just like the cells in cytokinesis.
Cytokinesis. Going back to the Latin roots, cyte means cell and kinesis means to move. This is finally when the cell splits. In animal cells, the cell pinches at the equator, creating what we call 2 identical daughter cells.
Let's celebrate! We have 2 identical cells!
And with the division finally complete, the cycle will repeat itself an infinite amount of times until it's told to stop.
Now that we know how the cell cycle occurs normally, we can look further into errors and their effects.
We all likely know someone who has been affected by some sort of cancer, and it's important to know where it comes from. Cancer is defined by cancer.gov as a disease in which some of the body's cells grow uncontrollably and spread to other parts of the body. Cancer has many contributing factors such as radiation, smoking, pollutants, chemicals, and viruses. Cancer can also be spread genetically. Cancer is the highest cause of death for adults younger than 65. Though a cure has yet to be found, there are many different treatment options such as radiation, chemotherapy, and surgery.
Many other experimental treatments are accessible to everyone due to the price, so wealthier patients may have a higher chance of remission or of being cancer-free. This unfortunately is not the only situation in healthcare where money can improve your treatment level. Some people will refuse treatment due to prices as well, which can cause a fairly treatable cancer to become terminal. Some people do believe that this is fair. However, I do not agree with putting a price on life maintenance. With this background knowledge of ethics and impacts, let's biologically break it down.
In cell division, we talked about checkpoints. In these checkpoints, there's a list that the cell needs to complete. Is the DNA replicated? Is the DNA damaged? And are there enough nutrients to provide the cell growth?
If the cell does not complete these checkpoints correctly, it'll do something called apoptosis, which is the cell dying off or killing itself. This also happens when genetic material gets funky. This most commonly occurs when the chromosomes get pulled apart in anaphase. When the chromosomes don't split at all or split incorrectly, it's called non-disjunction. The spindle attaches to a place other than the centromere, causing no splitting, incorrect splitting, or an uneven amount of chromosomes. Chromosomal instability often gets called the hallmark of cancer because of this. This can not only cause cancer, but also diseases such as Down syndrome.
This is not the only error that can cause cancer. Since we're on the topic of checkpoints, let's talk about proto-oncogenes, tumor suppressors, and DNA repair genes. Think of these genes as a car that drives us to the next stage. The proto-oncogenes are the gas pedals in cell division and code for growth factors receptors that signal the cell to move on. Tumor suppressor genes would be the brakes, making sure that the cell cycle doesn't move on until ready.
DNA repair genes are designed for combating errors in the second checkpoint. Sometimes the nucleus can send out abnormal orders to the cell, causing these genes to mutate. After a mutation in the proto-oncogenes, they become oncogenes, a cancer-causing gene. When these genes mutate, it is normally caused by a particular receptor called HER2, which are the receptors regarding growth factors, the biological molecules that affect cell growth and control proliferation or rapid cell number growth. When a mutation occurs in the oncogenes, the HER2 can allow the cell to overproliferate and become extremely sensitive to growth factors. Then you have a cell that is growing too quickly and ultimately dividing prematurely.
We know that a cycle means repeating, and so this mother cell with this particular mutation is dividing like crazy, which could create a neoplasm, also known as a tumor. Not only does it create a tumor, but with every division, the cell becomes more and more abnormal. This mass doesn't always have to be cancerous and sometimes can be benign, but ultimately this oncogene malfunction is one of the key errors that leads to breast cancer.
Benign means that the neoplasm has contact inhibition and will not grow out of control, making it non-cancerous. However, in a cancerous neoplasm, the mass continues to grow because of the lack of contact inhibition. The cells ignore their surroundings and continue to grow despite that lack of room. These cells, once a mass, often can create certain proteins that make vessels attached to them and provide them with nutrients to continue to grow.
Adding to the growing list of abnormalities, these cells can metastasize, which means that they can migrate to other parts of the body via your circulatory system. This is how cancer spreads to other organs. Normally, cells in our body stay where they were formed. They don't break off and go to other places to do jobs in other locations. They're formed in one spot, they are anchored there, and they stay there. Once cancer spreads to a place different from the original and is diagnosed, it's called metastatic.
So if a patient who originally got diagnosed with breast cancer gets a scan finding cancer in their lung, their cancer is called metastatic lung cancer. When a cancer metastasizes, it becomes more life-threatening. Cancer interferes with the daily duties of your organs, and when untreated, the organs can fail due to blockages, causing death.
As we said, cancer has yet to have a cure. Research is continuing daily in the race to find a cure. Though there isn't a cure, doctors have found functional treatment options that can send some patients into remission or even become cancer-free. Surgery, chemotherapy, radiation, immunotherapy, and stem cell therapy are some discovered therapy options.
Recent advancements include monoclonal antibodies for metastatic breast cancer, oncolytic virus therapy for melanoma, CAR T-cell therapy for lymphoblastic leukemia, and cancer vaccines for prostate cancer. Research and technology continue to develop for this disease, and doctors are hopeful that one day a cure may be found.
Thank you for joining us on our first episode of Biology Banter.
We hope you enjoyed listening and learning more about cell division errors in cancer.
But before we end, we need one more biology joke.
What is blood's message to the world?
I'm not sure.
Be positive.
I'm Sophia.
And I'm Lauryn, and this was Biology Banter. Since we recorded for over an hour, there's going to be bloopers, so here they are.
Sorry for interrupting.
I think we were growing apart, just like the cells in cytokinesis.
Cytokinesis. Going back to lactic acid.
Now that we know how the cell cycles...
Why'd you say it again?
The next word is our curse. And my brain's going too fast.
The cell cycle occurs.
Recent advancements include monoclonal antibodies for metastatic brain cancer.
Brain cancer.
For the country of Brants.
Brain cancer.
Recent advancements include monoclonal antibodies for metastatic brain cancer. I did it again!
Breast! I'll highlight